It looks good?
Yes.
Right. Perfect. Thank you. So my name is Monika Tutaj, and I'm research scientist around Genome database. And today I'm going to present some of the data from analysis of almost half of hybrid rat diversity panel strains, and which were sequenced and provided by Dr. Mindy Dwiner from Medical College of Wisconsin. So I will show my workflow, and with this I'll talk about the data. And so we received two data sets in 2019 and 2020. The quality of the data are pretty good, no problems with that. The only problem we have is like up to 19% of the duplication. And as we aim to have at least 30x coverage per strain, that could be problematic. But so as we are using GATK best recommended analysis workflow, there is optimization procedure in a covariance, as this was designed for human population variance analysis, which they usually have the problem with coverage too. So there are steps that are prepared for low coverage. So we'll see how it will work. So next, I will just show a couple of comparisons between two different rat genome references, as that was the main interesting part of this analysis, to compare the RN6 genome reference and the new one, which was just released in November 2020. And so we can see that there is a nice improvement in coverage with the new reference. And so nice improvement in the coverage. Still, there are some improper realigned pairs of reads and the chimeric reads, but nothing really serious or important. So in what is the critical step in variant call, we know that it's a base quality recalibration. So what does it mean? And usually from sequencing core, we receive the overestimated data. If you will look at this pink bar here, that's where they throw the data in 3 different bars. And what you want to use is a rat training set and adjust the quality score per base based on real data. And the data should look like a normal distribution. So basically all those 48 samples dropped in as quality score like 1 to 9 points. So it is one of the steps which eliminate false negative variance call in future. So the types of variants called between those two references, we can see increase in the SNPs and decrease in indels, and more details you will see in next slides. There are also the small fraction of multi-allelic SNPs and complex indels. Usually people don't put much of the interest in this, at least I didn't see many talks about those. And so to be valid variant, currently we are providing data which each variant has to pass all the 6 parameters. So these are stringent criteria, and we know so it's another way of minimizing false positive, and in the future we will need to adjust and rescue some of false negative results. So here I just compare the studies done over years, and major one which provided a single nucleotide variance data, and these are the 2013, 2015. RGD reanalyzed the 2013 data of the 25 to up to date to the recent rat reference. And currently we can see that the we've got 47 rat strains and a nice increase in population of single nucleotide variants. And also some comparison, like, improving of the softwares over the years. And also I will show some annotations, details. So the distribution of single nucleotide variants here, you can see that most of the samples show more than 5 million SNPs called. When we filtered 1 million, basically we are losing. Most of those variants are homozygous calls, but we can see that there's a significant percentage of heterozygous calls. And now this is really interesting because there are inbred rats, and where are those heterozygous calls are coming from? So I try to subset those data and look for the detailed information. And also you can see that there are a few samples, like these 4 samples are the descendant of the rat which was sequenced for the genome reference. There are also recombinant from brown Norway strain, and they are showing lower, generally lower numbers of variants. So if I will look in details for the heterozygous calls, and here now I'm showing comparison between both references, there are no much differences in generally, but there is a one thing here I wanted to point this. These heterozygous call are located, 5% of them, which are dark here green, are located in chromosome Y and unlocalized and unplaced contigs. So that's interesting. And that's also the reason that we've got the 18 megabase chromosome Y in a new reference compared with the previously assembled chromosome Y 3 megabase. But you could see here, I just next to the chromosome, I put this distribution of those heterozygous variants, how they where they are located. So just look at this brown Norway strains and the numbers I'm just showing here in the table. So what is here interesting, that these 2 strains, these are, as I mentioned before, 2 different data sets, 2019 and 2020. So these are the same strain, but sampled twice. And we can see that the other brown Norways, they generally compare between 2 assemblies, we've got a huge drop in the homozygous calls. So basically what we've got left to look closer at a brown Norway heterozygous variant call, which can help us resolve problematic regions still present in a new reference. Also, the number the variant call is much smaller than compared with between the assemblies. So now, looking at the insertion and deletions, you can see generally is a huge drop in a number of indels. So that's interesting, and also something to explore further. We can see that there is also some accumulation in the chromosome Y unlocalized, but then you could see that these are really heterozygous indels are taking over. The same as in SNPs. It is also more profound with the number of the homozygous calls. So we've got 5 times less in a brown Norway strains. So if we want to look at the functional consequences, so there's another part which we've got this gene model annotations. Yes, so currently we've got NCBI. I just put together all the assemblies available. You can see that we've got a nice increase in a gene over years, and the transcript numbers mapped Currently for NCBI, we've got more of an ensemble. For RN7, there is still available only rapid release, and we can see from our pipeline that each week this number are changing. So it's not still probably complete. And so that's why we use the NCBI annotation for doing the function consequence. This is done by SNP effect tools. Generally, there is no much of the differences. There is a slight difference in the high effect, high impact variants, SNPs, and drop in indels, which probably is also related to what we observed earlier. And we also run polyphen to prediction SNP, that's classified the variants as damaging. And so here's interesting also that we've got practically twice more for RN6 compared with the RN7. So now if we are talking about precision medicine, here's just example of Olga, which Jennifer mentioned, and this is my favorite tool when you want to play with the ontologist terms. So for example, disease, and I choose here the hypercholesterolemia. You've got the nice list of the genes here. And I want the intersection with coronary artery disease, and look at those just 41 overlapping genes in both diseases. And you can play also with chemicals if you're interested with some kind of the pharmacology studies. So these 41 genes, you can next put in choose some our toolkit tool, and I choose the variant visualizer. And by filtering through different criteria by location in a gene or the clinical significance, you can select, and this is for human, of course. So as we are starting with the human, and we are looking for the pathogenic variants. So more details you can see per variant. First, you've got numbers per genes. And next, we want to see what we've got in our population of rats. So we are choosing the different assemblies and put this the genes list in it. And the next step is choosing the strains you are interested. So here I'm showing, difference between RN7 and RN6, and right away I can see the nice difference, and that's interesting. I'll look at it, it's real, but if you see something like this, so there are two possibilities. So it's a genome problem or model annotation. So I need to look probably at the BAM files to see how the reads are falling there, if it's a nice alignment, and then the model, the gene model, how it was in the previous assembly, and to see why I don't see those data here. So for future work, that's what we need to finish the whole population of the hybrid rat diversity panel to start building the golden standard variants to have the final score about, how real is the variant, and also what we need to label which are not confident for users and researchers to know that, this is problematic because different reasons. So the model annotation or the still genome in this area is not finished. And so we uploaded those data on a download site. You can find it through this button here. There is a folder strain specific variants, and the project title is Dwinell MCW Hybrid Rat Diversity Program. And you can find both for RN6 and RN7 data sets, also divided by indel and variants. And here are all the files with index, so you can download them and research by yourself. And you can contact me if you will have any question, or you will need to rename the contigs, or I can provide you all the details. So in summary, I don't know if I've got the time for summary. In any case, what we are trying to generate this population of variants with confidence score. So to assist in finding appropriate disease model for this personal medicine, and the genetic variation in rat strains. So having the good set of variants is essential to build the high resolution associative maps and also to study complex traits. As we know, still I showed you the polyphen, but we need to implement more damaging like sift tools, as we want to see bigger picture, especially between strains, as it's not one gene. Yes, so basically you saw the HRDP portal presented by others, and so thank you.